Hello and welcome to my presentation. My name is Jonas Kuppler. I'm a postdoc at Ulm University, which is in southern Germany, and I'm mainly working on the effects of global change on plants, pollinators, their interactions, and those of course, on plant reproductive success and community changes. And today I will talk about how water deficit affects multivariate floral trait expressions, or the multivariate floral phenotype. And so this is quite interesting because, of course, floral traits are in the center of plant-pollinate interactions, and there are, on the one hand, mediate the interaction with the pollinators, and on the other hand, also affect the pollination efficiency, and those are key for successful plant reproduction. And it has been shown that actually water deficit changes floral phenotype expressions. So mostly it has been explored in terms of changes in the mean and of single traits. And so far the overall pattern is generally that the floral size in various measurements, as well as the number of flowers, is on average decreasing, simply because flowers are quite costly in terms of water, and these plants try to reduce water costs by reducing the overall floral area. And of course, these mean values, or the mean trait expressions, are of course quite important for the mediation of plant-pollinate interactions, but also it's not only the mean, it's also the variation of a trait or the variation of multivariate traits, as well as the integration of these traits. Integration simply meaning the correlation between traits. And these two parts actually have quite less explored, so we actually don't know if floral water deficit changes also the variation within species. So if, for example, on average within a species, there is less variation when the plant is stressed, or the traits become more closely correlated if there's, for example, stress. And this is what I'm going to talk about today. And we actually can have quite clear expectations what should happen under stress. So on the one hand, trait responses of the responses of intraspecific phenotypic variation and can follow the stress-induced hypothesis, which states that due to this developmental instability, as well as the release of cryptic genetic variation, there is an increase in intraspecific trait variation with stress, in this case, water stress. So the less water a plant have, the larger the variation should get. But there is also a hypothesis which says exactly the opposite, so the stress-reduced hypothesis, simply because under stress conditions, only specific phenotypes are actually favorable or suitable for the plant. And this is why interspecific variation decreases with stress level. So we have two outcomes which are possible, and of course all intermediate ones. And we can have similar expectations for integrations, meaning that integration or the correlation between traits can increase with stress level or also decrease with stress level based on the same ideas. So next to the absolute values of integration and variation, it's also actually interesting to see where these multivariate phenotypes placed in the trait space. So if a species comes to a new environment, then it actually usually needs to, well, coordinate a change of multivariate trait expressions to actually adapt to the new conditions. So in this case, for example, control to water deficit conditions. And depending how these two trait spaces, the trait space of the old and the new environment, are actually aligned to each other, it could be more easy for a plant species to adapt to the new conditions. So how? For example, if you have in the old trait space, you have a vector of maximum plasticity, so where the most variation is. And then if the new conditions in the new environment favor trait expressions which are aligned with this maximum plasticity, then it should be easier for a plant to actually change the expression to the new conditions. While if, like in this case, there is the vector is not aligned, where is a lot of variation possible in the old environment, then it could be less likely that actually plants can get to trait values or trait expression that are actually favorable in the new conditions. And this is a bit complicated. I mean, I'm also new to it, but I hope I have been not too imprecise. So what we did then, we actually collected data from already published studies. So data which were experimental studies, where there's one control group of a plant species and what experimental treatment where that's actually some kind of water deficit. And then if there are specific floral traits measured, so mostly size traits, so for example, floral size or nectar tube test, and kind of this. And then we also limited to minimum number of 30 individuals per treatment. And then we just calculated various indices of variation, trait integration, as well as the alignment. I will not go into details, it would cost too much time. And then we used the meta-analytic approach. So we calculated the log response ratio between the indices of the control and the water stress treatment, and then looked if we find any general patterns. So these are the results for the intraspecific variation, and there are different indices on the y-axis, and on the x-axis log response ratio. Log response ratio below 0 means that the water deficit treatment would have less variation than the control, and above 0 that the water deficit treatment would have higher variation than the control. What you can see here, of course, is that, well, there are mostly all around 0, so there is no differences in the variation, in interspecific variation to be precise, between control plants and water stressed plants. And there is kind of similar pattern for the integration, but we have two groups. So for some of the studies, there was actually no differences, but some studies showed a lower integration in the water deficit treatment. So overall, we can say we had these two hypotheses, so stress reduced and stress induced, that for the interspecific variation, well, it's most or less neutral, there is no differences, while for the trait integration, it kind of depends on the studies. And the interesting point here, so the studies that actually showed the lower trait integration were actually studies which really measured precise traits for pollination efficiency. So the other one often had like general floral size, but this one has specific ratios in there for relevant for the efficiency. And so this might be the case that actually, because there are specific the flowers were getting smaller in these studies also, and so if there is, for example, flowers getting smaller, but specific ratios have to be kept to allow efficient pollination, then actually it might be the case that integration actually decreases because the size and these ratios are not as much correlated. But this is kind of something we have to look into. Then now to the results of the shifts between the trait space or the alignment of the trait spaces. So in the first row, you can see that actually the maximum plasticity in new conditions within a study is usually quite well aligned with the mean plasticity in the old conditions, meaning there is only a small angle between these two vectors, which is kind of roughly here 5%. In the second row, you see the angle between the maximum plasticity vector of the new conditions and the old conditions, which is around 30%. So there's quite some differences between, but also still indicates that actually the same traits or the same trait groups are most variable in both conditions. And then for the last row, this is what I explained before. And here you can see that actually the vector to the new conditions is not very well aligned with the maximum plasticity of the old conditions or of the control in the water deficit. So this means that actually there might be some difficulties for plant species to adapt to these new water deficit conditions because the new trait space is not very well aligned with the maximum plasticity, so the traits where the most variation is harbored in the old conditions. So in the end, our results indicate that actually there is no differences in the intraspecific phenotypic variation between, in this case, well-watered and plants under water deficit, and that depending on the traits measured or included, there can be a difference in the integration, potentially depending if the plants needs to hold certain traits or trait ratios consistent to ensure pollination efficiency. And then also the alignment of the different varied spaces or the different plasticity vectors indicated that it might be difficult for some plants to adapt their flowers to the new conditions, but actually this is kind of speculative at the moment, and there need to be definitely more investigation around this, especially also because plants are also in the new conditions. They are also not only depending on water, but also depending on the planatus availability. So actually these multivariate phenotypes then have two selection pressures, abiotic and biotic ones. So, and in the end, I of course need to thank all my co-authors for sharing data with me and providing some inputs. And then if you have maybe also a data set which could be suitable, just get in contact. And otherwise, thank you very much for your attentions. And of course, I'm available in the chat for questions. Thank you.